Hi everyone, my name is Gaurav and I'm working as an HPC support specialist in the operations department of the Barcelona Supercomputing Center. I'm also working in the capacity of a research scientist in the Center of Excellence for Personalized Medicine. Today, it is an honor and a privilege for me to present before you that our team is doing to parallelize PhysiCell. We call this new parallelized version as PhysiCell-MPI or more lovingly as PhysiCell-X. So without any further ado, let us delve into the details. The first question which naturally arises is what is PhysiCell? PhysiCell is a physics-based multi-scale multicellular agent-based simulator which is capable of carrying out simulations in both 2D and 3D. It provides you with a stage which is a multi-substrate microenvironment as well as the actors which are the cells of the agents. The cells they are allowed to move, to grow, to divide. They can also die. They can interact biochemically or mechanically with other cells. They can release substrates into the microenvironment as well as ingest substrates from the microenvironment. PhysiCell can be thought of as being composed of two layers. The lower layer is called BioFEM, and the major function of BioFEM is to solve the partial differential equations modeling the diffusion processes after a finite volume discretization has been carried out on the domain. So the delta T diff, that is the time step for the diffusion processes, is the smallest time step in PhysiCell. The two other time steps, that is the delta T mechanical and the delta T phenotype, are taken care of by the second layer, which is PhysiCell itself. So it takes care of the movement of cells as well as the growth, the division, etc. As far as the software is concerned, it is completely written in C++ and it is shared memory parallelized using OpenMP. It enjoys minimum external dependencies and is open source. The latest version can be downloaded from the link which is shown over there. In general, PhysiCell is extremely efficient and you can carry out a 1 million cell simulation on approximately 4 to 6 cores. The biggest limitation of PhysiCell is that its execution is restricted to a single node of an HPC system. That is, the problem size that you can run is limited by the memory of a single node. Our aim in this particular project is to take away this particular limitation by enabling distributed memory parallelization in PhysiCell using MPI. Thus, our resultant code becomes hybrid, which is a combination of MPI and OpenMP. I'll talk about the parallelization of the base layer first, that is BioFEM parallelization. There are multiple kernels to be parallelized, and the first kernel that we parallelize is the domain partitioning kernel, and we carry out a 1D domain partitioning in the X direction. The reason why we carry it out in the X direction is because data is contiguous in memory in the X direction. As far as the microenvironment creation is concerned, it is fully parallel. That means all the MPI processes can simultaneously create a microenvironment. Coming to cell creation, the initial position generation of these cells is centralized. That means it is carried out by the first MPI process. But once these positions have been distributed to the other processes, the generation of cells, that is the agent creation, is fully parallel. Coming to the solver, the main purpose of BioFEM is to solve the partial differential equations which govern the diffusion of substrates. So the solver which is employed in BioFEM is the Thomas direct solver, which is a serial solver, and it is optimal in serial, and it is used for solving a tridiagonal system of linear equations. So an important point to note is that although we divide the domain in the X direction, but our solver in the parallel version is still serial. It is in the Y and the Z direction, our solver is optimal because we do not divide the physical domain in the Y and the Z direction. I'll talk at length about this particular solver. So this is our 1D domain decomposition scheme, and you can see that the physical domain is divided among 4 processes which are shown by 4 different colors, and we call this topology as 1 cross 4 cross 1, and as you can see that there are 4 processes. So like I mentioned, the lower layer must be parallelized before PhysiCell is parallelized, which is the second layer. BioFEM in itself is a full-fledged simulator or a library. So we have already been able to parallelize BioFEM, and we call it as BioFEM-X, and it is publicly available under the BSD3 clause license at the links given over there. In BioFEM, we if you want to scale up the number of walk cells, for example, we carry out a particular experiment with 0.5 billion walk cells and a single substrate, but the original version of BioFEM, which is only OpenMP parallelized, is unable to run this particular simulation because it runs out of memory, and we see an out of memory error. But BioFEM-X, that is the MPI plus OpenMP version, is able to carry out this particular simulation using 4 nodes and 8 nodes. And as you can see that there are significant advantages in multiple kernels, for example, like build microenvironment, Gaussian profile, definitely an advantage in file IO, because even with 4 nodes and 8 nodes, the timing almost remains the same in agent generation. But what you see is that there is no significant advantage in the source and the sink diffusion solver. I'll talk about that. I'll talk about this in a minute. If you scale up the number of substrate, then the original BioFEM is incapable of simulating approximately 400 substrates on a small domain size, which is 1500 cube. But BioFEM-X can simulate 400 or even 800 substrates by using just 2 nodes. Now we talk about the solver, so which is the bottleneck in our parallelized implementation. So we are currently in the process of replacing this particular serial solver with a modified version of the Thomas solver, which can be parallelized among multiple nodes. So if we consider a strong scaling problem of approximately 2.76 billion unknowns, we get a speed up of around 15, whereas the ideal speed up is 16 with 768 cores. Once the number of cores becomes 1536, you can see a significant loss in the efficiency. This particular loss in efficiency is because we are using a single node to solve a reduced system of equations. So right now we are also in the process of optimizing this particular solving of a reduced system of equations by distributing these reduced system of equations onto multiple nodes. If you consider weak scaling, then we started with approximately 2.7 billion unknowns and ran them on 48 cores, and we went up to 1.4 trillion unknowns on 24,576 cores, where we saw a loss of efficiency of about 40%. This loss in efficiency is because of the same reason which I just mentioned. The reading in red is an outlier and is not to be considered. We are also importantly bothered about the correctness and the completeness of BioFEM-X. So over here you see the initial concentration of 3 substrates and the final concentration of 3 substrates in serial as well as parallel. And this is for a complete 3D tumor growth example. And what you can see is that the 2 plots are identical. So there is no difference between the serial and the parallel simulation, hence verifying the correctness of our implementation. Coming to PhysiCell parallelization, there are once again multiple kernels to be parallelized, for example, the cell division. So a significant difference in the parallelized version and the serial version is that once cell division takes place, we do not allow the parent or the daughter to move out of the subdomain. The cell movement across subdomains is right now a heavyweight process, and we are packing a lot of information which describes a cell in the other subdomain. So once a cell passes from one subdomain to another subdomain, it has to be destroyed in the first subdomain and created in the other subdomain. As far as velocity update is concerned, it is a very computationally intensive process. And if you consider a local velocity update, then a particular cell only takes into account the cells which are in its current walk cell. But for a remote velocity update, it also considers the cells which are in its 26 neighboring walk cells. The output file parallelization is also an extremely time-consuming process, but we have already parallelized the SVG and the MATLAB file writing process. So I show a predator-prey simulation. There are 50 preys which run from predators. To prevent the preys from crossing the boundaries of the domain boundaries also act like predators. So the predators produce some kind of a smell which the preys detect and run away. So the challenging part about this particular simulation is that you see a large, fast movement of cells across the cell, the subdomain boundary. This is the simulation. The blue cells are the preys. The yellow cells are the predators. And now the simulation repeats. This is a very, very small scale simulation. So we record the number of living cells, the apoptotic cells, the necrotic cells at various times in the serial and the parallel simulation. And as you can see, the plots are very, very similar. But once again, we are devoting efforts to check the correctness of the simulation. Now we can scale up this particular simulation to about 1 million preys, 0.1 million predators, and by increasing the domain size. PhysiCell-X, which is PhysiCell-MPI, was run on 10 nodes or 480 cores. This is the simulation. So we initially start with about 1.1 million agents or cells, which grows up to about 9.5 or 9.6 million agents or cells. The diffusion time step is 0.01, the mechanical time step is 0.1, and the delta T phenotype is 6, and this represents approximately 36 minutes of simulation. And now the simulation keeps repeating. If you compare the serial time versus the parallel time, the serial simulation takes approximately 177 hours, which is 7.4 days wall clock time. The parallel simulation takes only 43.5 hours, which is just 1.81 days. So we obtain an approximate speed up of 4.06. You save approximately 5.5 days, and we see the advantage of PhysiCell-X over PhysiCell-MPI. So in summary, we are developing a distributed parallel version of PhysiCell-X. Right now, we have 1D domain partitioning, and we aim to simulate billions of cells. There is a lot of work to be done. So we need to replace the diffusion solver. We need to consider 3D domain partitioning, and just like with any other software, we need to carry out more testing, optimization, and possibly a redesign. These are some of the references. Thank you very much for your time. And if there are some questions, we would be very, very happy to answer those questions. Thank you.